Good day, dear juniors. Welcome to our first topic for finals. Today, we will talk about the hemocytometer and the RBC or the erythrocyte count. I am your speaker for this module, Ms. Sophia Alexis Derit, so let's get started. First, let's talk about the different learning objectives. At the end of the laboratory exercise, the students must be able to number one, understand the underlying principle of cell counting using a hemocytometer and how it relates to determining cell concentration and viability. Number two, familiarize yourself with hemocytometer components, the various parts including the counting grid, counting areas, subsquares, and the overall design. Number three, understand the correct techniques for handling and using a hemocytometer. Number four, develop the ability to calculate cell concentration and viability based on the cell counts obtained using a hemocytometer. Number five, differentiate the different types of hemocytometers, their features, and when to use each type based on specific cell counting requirements. Number six, explain the principles and procedures involved in the different methods of cell counting. And lastly, number seven, discuss the importance of the RBC count in the diagnosis of hematologic diseases. First, let's talk about the principle of cell counting. So, the principle of cell counting using a Neubauer counting chamber, the process starts with diluting the sample. This step ensures that the cell concentration is within a range that makes counting manageable and accurate. Too concentrated, and it becomes difficult to distinguish individual cells. Too dilute, and there might not be enough cells to count reliably. Next, we load a known volume of this diluted sample into the counting chamber. The chamber has a precise grid etched into it, allowing us to count cells within specific areas under the microscope. Once the sample is loaded, the actual counting begins. We observe and count the cells within defined grid areas, which can then be used to calculate the concentration of cells in the original sample. The accuracy of this technique relies on both the correct dilution and precise counting. It is important to note when it comes to the different principles of cell counting, or when it comes to the different cell counting, such as the WBC count, the RBC count, and the platelet count, they all use one principle. They will be only differentiated based on the dilution factor, and the dilution factor is brought about by the different dilution pipettes that we use for each procedure. Next, manual methods are necessary when counts exceeds the linearity of an instrument, non-functional instruments and no backups available, remote laboratories, and disaster situations when testing in the field. Again, since all our methods are already automated, it is very rare for a laboratory to perform manual methods. Still, it's important for students to know the basic principles behind these methods. So while automated systems are commonly used for cell counting, manual methods still play a crucial role in several situations, as I mentioned earlier. So, for example, in remote laboratories where access to advanced equipment may be limited, manual counting remains a vital tool. Additionally, during disaster situations, when testing is done in the field, manual methods offer a practical solution for obtaining immediate results when resources are scarce or compromised. So next, let's talk about the hemocytometer itself. So the hemocytometer is described to be a thick glass slide with grids that are used for counting. The most common type is the Levy chamber with improved Neubauer ruling. Hemocytometers and cover slips must meet the specifications of the National Bureau of Standards, as indicated by the initials NBS on the chamber. So as you can see, the hemocytometer, it is composed of two raised surfaces, each with a 3 mm by 3 mm square counting area or grid, with a total area of 9 mm squared. It is separated by what we call an age-shaped moat. So it is made up of nine 1 millimeter by 1 millimeter squares. Each of the four corners wherein the WBC is counted, or for each of the four corner squares, is subdivided into 16 squares, and the center square subdivided into 25 smaller squares. Each of the smallest squares is 0.2 millimeter by 0.2 millimeter, which is 1 of 25th of the center square. So let's get more closer to the picture of the height of the hemocytometer. So again, the counting chamber is made up of 9 1 millimeter by 1 millimeter squares. So when we say the 9 squares, this is what I mean. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So again, we have 9 squares. And each square, the size of each square is 1 millimeter by 1 millimeter. Here. So this square is made up of 1 millimeter by 1 millimeter, again 1 millimeter by 1 millimeter, and again it is made up of 9 squares. Next, in this area, we can see that there are 4 corner squares. So when we say 4 corner squares, we mean this one, this one, and this one. Corner or dulo. Okay? So these corner squares is where we count our white blood cells. And for each corner square, you can see that it is subdivided into 16 squares. So again, we have 4 corner squares, these ones, and then we can see that they are further divided into 16 squares. We have 1, 2, 3, 4 by 4, therefore 16. Each corner square is divided or subdivided into 16 squares. Next, we have our one large central square. For the one large central square, it is subdivided into 25 secondary or intermediate squares. So we can see that this is 1, 2, 3, 4, 5, and then 1, 2, 3, 4, 5. 5 by 5 or 5 times 5 is equal to 25. So again, the one large central square is subdivided into 25 secondary intermediate squares. So this area is where we count our red blood cells. Allow me to repeat the whole part that I just said. So again, in our counting chamber, we have 9 large squares. In each square, it is sized up to 1 millimeter by 1 millimeter. For the 4 large corner squares, they are further subdivided into 16 squares. And in those 4 large corner squares is where we count our WBC or our white blood cells. Next, we have a central large square which is further divided into 25 intermediate or secondary squares. So here we can see that this is where our red blood cell is counted. So I mentioned earlier during the discussion of the principles that the cell counts, they have similar principles and they will only differ in the dilution factor brought about by the dilution pipette. Another thing that will differentiate the different counting methods will be the area in which they are counted, the area within the hemocytometer. So again, this is where our red blood cell or our white blood cell is counted, and in the middle is where our red blood cells are counted. Just to add, when using a hemocytometer, a cover slip is carefully placed on top of the counting surfaces to form a defined space between the cover slip and the grid. So the distance between the counting surface or the grid and the cover slip is precisely zero point 0.1 millimeters. Okay? Again, do not be confused. The distance between the counting surface and the cover slip is precisely zero point 0.1 millimeters. This distance is critical because it determines the depth of the chamber, which is necessary for calculating the volume of the sample being counted. So each grid or counting area on the hemocytometer is composed of multiple squares arranged in a specific pattern, as we talked about earlier. So since the distance between the grid and the cover slip is again zero point 0.1 millimeter, and we know that the area of the grid's surface, we can calculate the volume of the chamber above the grid. So for one entire grid, just like this one, the area is typically 9 millimeters squared. Bakit po siya 9? Because this is 1 by 1, 1 by 1, 1 by 1, and we have total of 9 squares. So for one entire grid, the area is typically 9 millimeters squared. So that is a 3 by 3 millimeter grid. So multiplying the area by the depth, which is again zero point 0.1 millimeter, gives us the total volume above the grid. So we multiply the volume is equal to 9 millimeters squared times the depth, which is zero point 0.1 millimeter. Therefore, we get zero point 0.9 millimeter cube. I hope that you get that. So this volume represents the total space filled by the sample above the grid. So tinitingnan po natin dito is yung space doon sa gitna ng cover slip and ng counting chamber. And this is only on one side of the hemocytometer. Again, I said earlier, we have two sides of the hemocytometer. This is one side and this is the other side. This is the cover slip and this is what it looks like when it is on the side view. Ayan, no? So we can see that the cover slip and this is the counting grid that there is zero point 0.1 millimeter distance. Okay, so that is what we multiply with 9 millimeter squared. So understanding this volume is crucial because it allows us to calculate the concentration of cells in the original sample once we've counted the cells within this defined area. Don't worry if you are still confused with this part. Once we meet during our face-to-face, during our pre-lab discussion, I will discuss this further. Okay, so next let's talk about the different types of hemocytometers. Number one, we have the old Neubauer counting chamber. So this has improved grid lines and markings that make it easier to count cells or particles accurately. Next, we have the Nageoet counting chamber. So this manually counts low leukocyte concentrations below the capabilities of a standard hemocytometer. This contains a counting area containing 4 rectangular areas. My apologies, I think this is pronounced as Najot. So it is French, Najot counting chamber. Next, we have the Malassez counting chamber. So this is commonly used for counting cells in the cerebrospinal fluid or the CSF or for counting nematodes. And this one has a double net ruling. Next, we have the Berker counting chamber. So this has a smaller grid pattern and it is used for counting smaller cells or particles or red blood cells. Next, we have the Fuchs Rosenthal counting chamber. It is designed for counting leukocytes, eosinophils, and cells in the cerebrospinal fluid. Grid patterns is etched directly onto the glass surface, and the chamber is zero point 0.2 millimeters deep. Okay, now let's talk about the two different dilution pipettes. Let's compare them side by side. So here on the left side, we have the RBC pipette, and on the left side, we have the WBC pipette. Take note that, of course, if we are using or if we are going to perform an RBC count, therefore we are going to use an RBC pipette and vice versa. So to compare it side by side, as you can see, The RBC pipette and the WBC pipette, they both have a rubber tube, this one. And then they also both have a mouthpiece. But take note that we do not use our mouths to pipette using these pipettes. Instead, we use a aspirator. Next, as you can see, the markings here in the first or in the top are different. For the RBC pipette, we have a 101 marking here. As you can see here, it is written 101 or 101. Next, in the WBC pipette, you can see that the marking is 11. So those are two major differences that we can see between the red blood cell pipette and the WBC pipette. Next, you can see that this one here in the red blood cell pipette, there is the presence of a red bead. Okay? Meron pong red bead inside our pipette. And here in the WBC pipette, we can see that there is a white bead here. Next, both of them have a 1 unit here. So it's the same here. By the way, the bead is used to it is used for mixing the sample, because here in the pipette, this is where we put our sample or the blood and also the diluting fluid. So this is where we mix the sample, and then this is where the sample comes from when we put it inside the hemocytometer. Ayan. And then here, we also have a zero point 0.5 mark or zero point 0.5 unit. So it is just the same. So the major difference between the RBC and the WBC pipette is the first unit here. This one, it has a 101 unit, and this one, it has a 11 unit. And again, we have the red bead and the white bead. Okay. So let's talk about more of the RBC pipette. So for the RBC Thoma pipette, if our dilution is 1 is to 100, we fill up the blood up until the 1 mark. We fill up the blood up until the 1 blood or the 1 mark. So we fill up the blood up until here. And then the dilution fluid is up until the 101 mark. That is again if the dilution is 1 is to 100. Next, if our dilution is 1 is to 200, what we do is we still fill up the blood up until the 0.01 or 0.05 mark. We fill up the blood up until the 0.05 mark, and then we fill up the dilution fluid up until the 101 mark. Again, let me repeat. If our dilution is 1 is to 100, we fill up the blood up until here at the 1 unit mark. And then we continuously fill up the dilution fluid up until the 101 mark. Again, that is if our dilution is 1 is to 100. Next, if our dilution fluid or if our dilution is 1 is to 200, we fill up the blood up until only here in the 0.5 unit mark. And then we continuously fill up the diluting fluid up until the 101 mark. That is when our dilution is 1 is to 200. Next, for our WBC pipette, again, this is a white bead. Another thing that we have to notice that it has a smaller bulb and a larger lumen. Yan. So if our dilution here, it is a bit smaller compared to our white or compared to our red blood cell, if in the WBC Thoma pipette, if the dilution is 1 is to 10, we fill up the blood up until the 1 mark here, and then we fill up the dilution fluid up until the 11 mark. Again, if our dilution is 1 is to 100, we fill up the blood up until the 1 unit mark. and then we fill up the dilution fluid up until the 11th mark. Next, if the dilution fluid or if our dilution is 1 is to 20, we fill up the blood similar to the procedure that we did for the red blood cell count. We fill up the blood only up until here in the zero point 0.5 mark. And then we continue filling it up until the 11th mark. Again, that is when our dilution is 1 is to 20. So for the red blood cell count, the standard dilution for RBC count is 1 is to 200. So again, if it is 1 is to 200, how do we do it? So we just fill the blood up until here and then the dilution fluid up until here. So the RBC diluting pipette can perform higher dilutions, obviously, and for the WBC count, it only performs up until 11 units. So those are the major differences between the red blood cell pipette or the red blood cell thoma pipette and the WBC thoma pipette. Okay. So for standardized pipetting and diluting of blood and other body fluids for increased safety, accuracy, and precision, we can use the Unipet system. So for the different parts of the Unipet system, number one, we have the reservoir. So for the reservoir, it contains pre-measured volume of diluting fluid and is sealed by a thin covering of plastic called the diaphragm here. So this is at the neck. Next, we have the pipette. This is self-filling and it is available in various sizes depending on the procedure. It is color-coded according to size, and the end tip of the opposite side is called the overflow chamber. Sorry, it is not really shown here in the picture, but this is the overflow chamber. And then we have the pipette shield. So obviously, it protects the pipette. And this is also utilized to puncture the reservoir diaphragm prior to use. So in our experiment, we won't be really using a Unipet system. No, we will be using the manual methods in using or in diluting our sample, which is the RBC pipette and the WBC pipette. So good to know lang naman ito. Okay? So for the different rules for handling and using a hemocytometer, so of course, to ensure accurate results when using a hemocytometer, it is essential to follow few key rules. So first, what we do is we load the chamber with a suitable volume of a well-mixed sample. So this helps prevent errors caused by uneven distribution of cells. After loading, it is important to let the sample settle for a few minutes to ensure the cells spread evenly across the counting grid. So allow me to reiterate that it is important that we leave it alone. Now we leave the samples to settle for a few minutes before we can count them. Additionally, maintaining a moist chamber during this step helps prevent the sample from evaporating, which could, of course, affect the concentration of cells. So once the sample is ready, cells are counted in a designated area of the grid using an appropriate magnification, usually 10 or 20, depending on the cell type and concentration. So again, just for review, in our 9 large squares, we count our WBCs on the 4 large corner squares, and we count our red blood cell in the middle large squares, which is further divided into 25 secondary squares. So for the moist chamber, just to describe it, it is made by placing a piece of damp filter paper at the bottom of a petri dish, an applicator stick broken in half so it can serve as a support. When counting cells using a hemocytometer, again, it is important that we follow specific guidelines to maintain consistency and avoid counting errors. So one key rule is how to handle cells that touch outside the boundaries of the large square you are counting in. So remember, class, that we only count cells that touch the left and top outside lines of the large square. So this rule is designed to avoid double counting cells. So it is called actually the inverted L. This one. Sorry. Ayan, dito lang. So the left and top, as you imagine, it is like an inverted L. Okay? So the reason why we only count the squares that touch the left and top side, again, this is to avoid the double counting of cells. If we counted cells that touch all four sides of the outside lines, the same cells might be counted again in the neighboring squares when we move to the next counting area. Imagine when you move the slide papunta sa left, or papunta sa right, mabibilang mo na naman yung binilang mo kanina. So it will, or double counting of cells might occur. So by counting only the cells that touch the left and the top lines, and ignoring those on the right and bottom lines, we ensure that each cell is counted once and only once. So this approach maintains accuracy across the entire grid and prevents overlap in the counting process. So to continue, cells that touch the outside lines of the large square, again, we count only that touch the left and the top side if we imagine that this is an inverted L. So chambers with double lines disregard that touch the right and the lower sides of the outside margin. For the pattern, we can use what we call the serpentine method. It is like a snake in counting or in moving our slide. Next, for the calculations, it is important that we know the following volumes, such as the percent of cell viability, which is the total viable cells multiplied by the unstained cells divided into the total cells, which is multiplied by the sum of viable plus the dead cells, and then multiplied by 100. So the following factors or the following solutions are given to you, you can go ahead and screenshot this for your reference. Again, we should be able to identify the percent of cell viability, the average viable cell count per square, the dilution factor, the viable cells per millimeter, and the total viable cells per sample. For the next following slides, we will be giving sample calculations for your practice. So first, let's identify the cell viability. So given our formula and given our values, we can go ahead and calculate these. So the total viable cells, which is 54, which is unstained, and then we divide it by the total cells, which is 57, or the total number of cells. And then it is divided, or it is multiplied by 100. Therefore, we get 94.7. Next one, the average viable cell count per square. So we simply get the total viable cells. That is, again, 54. We have the total viable cells divided by 4. Therefore, we get 13.5. Next, for the dilution factor. So for the dilution factor, this is not always constant, no? Because again, it will differ depending on what we are trying to identify. But let's say, for example, our volume sample, or for example, we are trying to identify WBC count. So we are going to use the volume of sample, which is 50, and the volume of diluting fluid is also 50. If you want a reference from this, you can go ahead and read your protocol. So that is 50 plus 50, it will equal to 100 microliters divided into the volume of sample, again, which is 50. So 100 divided by 50 is equal to 2. So our dilution factor is 2. Next, the viable cells per millimeter. So the formula is the average viable cell count per square multiplied by the dilution factor, which we got earlier, times 104. So the total, or the average viable cell count per square, which is obtained earlier here in letter B, that is 13.5, multiplied by the dilution factor, which is what we got here, and then multiplied by a constant, which is 104. Therefore, we get 270,000 cells per millimeter. And lastly, we have the total viable cells per sample. So it is equal to viable cells times the volume of sample before dilution. So 270,000 times, again, kanina, I said that our sample volume is 50 microliters. And if we convert that into ml, we get zero point 0.5 ml. Therefore, we get 270,000 times zero point 0.5 ml, we get 13,500 cells per sample. So that ends the first part of our discussion. Kindly view our next video for the next part of the discussion. Thank you so much.